Hello, everyone. My name is Chan-Seok Jeong. I'm going to talk about a transformer-based method for predicting anti-CRISPR function. Anti-CRISPR is a bacteriophage gene that inhibits the bacterial CRISPR-Cas system and interferes with the bacterial immune function. It is also highly likely to be utilized as a natural switch to control CRISPR-based gene editing technology. For this, it is important to discover new anti-CRISPRs, and computational technology can be utilized for efficient screening. However, as the existence of anti-CRISPR has been revealed relatively recently, there are still not many experimentally verified data. This makes it difficult to develop effective artificial intelligence approaches. As a previous speaker mentioned in detail, Transformer is an artificial intelligence model of an encoder-decoder structure and operates based on an attention mechanism, unlike conventional RNNs. Transformer has been used in various fields since it was developed in 2017 and established itself as the most popular technology in the field of natural language processing. The most widely known transformer model, BERT, uses a pre-trained language model and subsequently is fine-tuned for a variety of natural language processing tasks. In the same way, a similar artificial intelligence can be constructed by replacing natural language with biological sequences. The type model developed in 2019 is a transformer-based model that uses an amino acid sequence as an input and can be fine-tuned to various bioinformatics tasks. In my work, I developed a predictive model that recognizes anti-CRISPR proteins by adding a classification layer to the transformer model pre-trained with unlabeled protein sequences and fine-tuning it with labeled data, in this case, anti-CRISPR data. For training and testing the prediction model, I collected CRISPR and non-anti-CRISPR protein sequences from Anti-CRISPR DB and Phaster DB lists. The data filtering was performed to construct sufficiently verified anti-CRISPR and putative non-anti-CRISPR data. The clustering was performed at 40% sequence identity to avoid data bias. Hyperparameters and model architecture of a classification layer for fine-tuned model was selected through 5-fold cross-validation. This is the validation result. And when the transformer-based model was tested, the AUC of the transformer model was 0.827, which was improved by 22.9% compared to XGBoost model AUC. In addition, the sensitivity of the transformer model was improved by about 2.1 times compared to the conventional method at a specificity of 0.95. Consistently, the sensitivity of the transformer model were improved by more than 60% at specificity of 0.99 and 0.90. The performance difference according to the anti-CRISPR classification was compared. The transformer model outperformed the existence method for almost all inhibited CRISPR-Cas types and anti-CRISPR families. In particular, the sensitivity to ACR2A7 and ACR2A9 families increased more than twofold. Those families inhibit CRISPR-Cas9 and are known to be widely distributed in various bacteriophage phyla. This suggests that the transformer model is effective in recognizing functional associations between evolutionarily distant genes. Consequently, transformer-based anti-CRISPR precision method developed through this study is more effective than the existence method. In particular, it is expected to be usefully used for genome investigations because any additional information is not required other than sequence information. I close my talk by thanking the team members for their help with this work. Thank you for your attention. May I take your questions?